Hello, everyone. Hello, great students. Hello, powerful students. Hello, brilliant students. Hello, beautiful students. Hello, handsome students. Hello, great future scientists. Hello, great future politicians. Hello, great future administrators and key policy makers both in Nigeria, Africa, and the whole world. Today, we are looking at the subject of methods in microbial genetics. Methods in microbial genetics. Already you have been introduced to microbial genetics and you have seen one or two things in microbial genetics. You've seen the codons, you've seen many other things in microbial genetics. Now we want to look at the methods that are used in microbial genetics. I must say from the start that the methods used in microbial genetics are many. The methodologies are many. Nobody can give you all the methods because as we are looking at the method that have been used before and are still being used now, new methods are being developed. The developed ones are being modified. So we cannot completely cover everything. So I will advise on your own, go ahead and read and make more research and get more details about the subject matter we're looking at. Before we go in depth into the methods, I want to talk about some genetic tools and techniques. The number one genetic tool and technique I'm going to tell you about is DNA microarrays. DNA microarrays. What do you mean by DNA microarrays? A DNA microarray or DNA microarrays enables scientists to monitor the activities of hundreds or thousands of genes at once. Scientists can monitor the activities of hundreds or thousands of genes at once. All microarrays, also called DNA chips or gene chips, work on the basic principle that the complementary nucleotide sequence in DNA and RNA match up. Definitely you've known about complementary sequence now, complementary nucleotide sequence. So that when you have A and the complementary is what is T, and when you have C, the complementary is G. So if a microbiologist is studying bacteria that bioremediate or that biodegrade or break down toxic waste and wants to know which specific genes are active when that bacterium is degrading, say PCBs, that is chlorinated biophenol, he will likely use a tool called the DNA microarray. Now, another tool is serial analysis of gene expression, which is also called SAGE. Serial analysis of gene expression, or SAGE, is an experimental technique designed to gain a direct and quantitative measure of gene expression. You can see that DNA microarray is a kind of indirect because the principle is based on complementarity. But in the case of SAGE, there's a direct and quantitative, not just quantitative measure of gene expression. The SAGE method is based on the isolation of unique sequence tags. 9 to 10 base pair in length from individual messenger RNAs, and concatenation, the action of linking things together in a series or the condition of being linked in such a way of tags serially into long DNA molecules for a lump sum sequencing. I already shared with you the material on Sanger sequencing, and you see that Sanger sequencing is a chain termination sequencing, because when sequencing starts, it cannot just continue and not ending. So what in sequencing, you sequence this, the DNA is broken into pieces. Then when you start sequencing, you stop at the point, the chain terminates. That sequence may contain 50 base pairs. Another one, the chain terminates in the same reaction mixture, may contain 60, another one 70, another one 45. At the end of the day, everything will be put together, will be arranged together so that you can get maybe 1,000 base pairs, 500 base pair, 700 base pair, depending on the primers you are using. Your forward and reverse primer will determine the length of the base pair of the nucleotide sequence you're going to have. So in the case of serial analysis of gene expression, there is a isolation of unique sequence tags, 9 to 10 base pair in length from individual messenger RNA, and then arranging them together in a series, and then reading what you have as a lump sum sequence. Another genetic tool is 16S ribosomal RNA. Scientists also use molecular tools to extract and compare bits of a particular kind of RNA from samples in order to determine if previously known or new microbes are present in a particular environment. This technique is widely used as a biomarker and for microbial ecology studies. It uses a particular kind of RNA known as 16S ribosomal RNA or 16S RNA. 16S RNA is useful for phyletic classification because it occurs in all bacteria and it contains highly conserved stable sequences. In fact, the main reason is because it contains highly conserved stable sequences. There are other types of RNA, but the 16S have been found to contain highly conserved stable sequence of nucleotides, as well as more variable sequences, so that it permits classification at both ends of the taxonomic spectrum. Thus, divergence at the level of higher taxonomic ranks, e.g. kingdom, domain, may be evident when comparing the highly conserved sequences of different organisms, while divergence of the strain or species level may be seen when comparisons are made of the more variable regions. 16S ribosomal RNA analysis is also used at lower taxonomic level. For example, in a given genus, the 16S RNA of different species have been found generally to differ by at least 1.5%. However, certain species of Bacillus, which are clearly distinguishable by DNA hybridization, were shown to have virtually identical 16S RNAs. So what this is saying is that previously we came up with 16S RNA and we said that was the best. Today now we have DNA hybridization. We have different, apart from 16S RNA of different organisms of the same organism. We are talking about the plate count anomaly. The plate count anomaly, the problems of plating and the counting on plate and identifying on plate. You can see the same colony, the same type of colony on the plate, but they are different organisms and there are anomalies. That was why we move on to 16S RNA. Now 16S RNA, even for Bacillus, they may have the same property, whereas they are of different species. So we are saying that 16S RNA alone may not be able to even identify everything. There's also DNA hybridization. There are other tests that you have to do to be able to buttress this 16S RNA, despite its being having highly conserved sequences. Now we're going to stop on that for now and move on to another genetic tool, another technique, which is the gene sequencing. The advent of PCR has led to an explosion of microbial gene sequencing in recent years. PCR spells out the entire sequence of the nucleotide bases, the A's, the G's, the C's, and the T's in a DNA molecule that code for a specific protein. Scientists also use sequencing to spell out some start to end every single nucleotide in an organism's DNA, that is the entire genome. And we have today what is called whole genome sequencing, where you sequence the whole genome of the organism. And a lot of work is going on this area, and I want you students to key into this area because it is the future of microbiology. Now I've done another material on Sanger sequencing which I sent to you. I was thinking we're not going to take it up, but we're going to take it up now, because you could see this is one of the tools used in genetics. So that material on Sanger sequencing, take it seriously, because we are going to be judged based on that material. When I say we are going to be judged, we are going to answer questions on that material. So take the Sanger sequencing material seriously. It is part and parcel of what we are covering here. The next genetic tool is PCR. Not all microorganisms can be grown in culture. I've told you about the plate count anomaly. You want to isolate organisms in the air from an environment. You want to isolate organisms in water, let's even say in air, or from water, or from soil, from petroleum, or from wherever. You want to isolate it on a plate. You understand? You want to isolate those organisms on a plate of PDA or nutrient agar or other such medium. When I say plate, I mean petri dish. Now, is it possible for you to get all the organisms in the water on nutrient agar? Not all of them will grow on nutrient agar. Not all of them will grow on any type of agar. Some organisms will not still grow. On PDA, some fungi will not still appear. While some appear, some will not appear. So that is why we have the plate count anomaly. In fact, scientists and research have shown us that about 95% of organisms that ought to appear will not appear. And that gives us the plate count anomaly. So not all microorganisms can be grown in culture. In fact, microbiologists have figured out how to grow very few species in the lab. Less than 1%. I was even saying 95% cannot be grown. Less than 1% of all microbial species in the world. PCR allows scientists to extract and analyze bits of microbial DNA from samples, meaning they don't need to find and grow whole cells. PCR is an essential element in DNA fingerprinting and in the sequencing of genes and entire genomes. So go and find out the meaning of DNA fingerprinting, because it's going to come up as part of your questions in this course. What is DNA fingerprinting and how does it link up with PCR? Now, what is even PCR? PCR means polymerase chain reaction. Polymerase chain reaction. So go and read more about PCR and the principle of PCR. And PCR started in 1985 and it was developed by Kerry Mullis. It was developed by Kerry Mullis in 1985. Now we'll go on again to our method in microbial genetics. The number one thing we want to talk about now is gene transfer. Gene transfer. Student transfer from one school to another school. People transfer from one department to another department. They call it movement during admission process. Genes can also be transferred. Bacteria reproduce by the process of binary fission. And I was talking about, by now you should know what we call doubling time and generation time. That is the time it takes for bacteria to divide into 2, and then 2 divided into 4, 4 divided into 8. So if you look at that time that bacteria uses to divide into 2 by binary fission, if the doubling time of a bacterium or generation time of a bacterium is 20 minutes or 30 minutes, I think you can estimate, you can ask you the question, how many cells are we going to have in 24 hours, or in 1 day, or in 2 days, or in 3 days, or even in a week. So we don't even need in a week if it's bacteria. All we just need is in 24 hours. So if the doubling time of a bacterium is 20 minutes, how many of that bacteria are you going to have in 24 hours? So note it down. It's likely to also show up on the day of reckoning. So Bacteria reproduce by the process of binary fission. In this process, the chromosome in the mother cell is replicated and a copy is allocated to each of the daughter cells. As a result, the two daughter cells are genetically identical. If the daughter cells are always identical to the mother, how are different strains of the same bacterial species created? The answer lies in certain events that change the bacterial chromosome, and then these changes are passed on to future generations by binary fission. Some of the events that result in heritable changes in the genome are genetic transfer and recombination, plasmids, and transposons. Now, back to the question on generation time. Bacteria reproduce by the process of binary fission, and the generation time of some bacterium are 20 minutes, 30 minutes, or even less time. Generation time is also called doubling time. And the question I want to ask you, which I want to drop the answer in the comment section below, is that if a bacterium, if the doubling time or generation time of a bacterium is 20 minutes, if the doubling time of a single bacterium is 20 minutes, how many of it are we going to have in 24 hours? Calculate it and write the answer in the comment section below. Now, we've talked about gene transfer. Now let's talk about recombination. I want you to follow me step by step so that you can understand these terminologies and this subject matter. Genetic recombination refers to the exchange between two DNA molecules. It results in new combinations of genes on the chromosome. In crossing over two homologous chromosomes, chromosomes that contain the same sequence of genes but can have different alleles, they break at corresponding points, they switch fragments and rejoin. It's like you have a pencil, and then the pencil is broken into two. The pencil is made up of the same thing, the same material. You break into two and then rejoin it together. When you are rejoining together, some particles of the pencil are falling off. Because maybe the pencils fell to the ground, some particles in the air has also joined to the part where you broke the pencil from. So when you are joining the pencil back together, you think that it's the pencil that I'm just joining together. No, some parts have been lost, some parts have been added. I think you get it now. So in the case of microbial, in the case of genes, those little fragments are important, and that little change can cause a little change, and that little change can make it to be different from the original organism from which it was broken from. You broke the pencil, you put it together. When you broke it, some parts of the wood used in making the pencil are falling to the ground. You cannot pick those ones. They cannot even be visible. Some particles, some atoms are falling from it. In the process of combining together, some particles from air, maybe dust, maybe soil, maybe sand, maybe anything, will also rejoin together. So that pencil, you think it is original pencil, it is a different pencil entirely. I think you get it now. So this is, I'm talking about recombination. I'm using that pencil as an example so that you understand it clearly. Anywhere you go, you'll be able to understand what I'm saying. So I said in crossing over two homologous chromosomes, like a pencil, chromosomes that contain the same sequence of genes but can have different alleles, break at corresponding points or switch fragments and rejoin. So you have two pencils, pencil A, pencil B, complete. You break pencil A, you break pencil B at the same point. You now join the other part of A to the other part of B, the other part of B to the other part of A. The pencils are the same initially. Now, what is going to happen? That is recombination. The result is two recombinant chromosomes. In bacteria, crossing over involves a chromosome segment entering the cell and aligning with its homologous segment on the bacterial chromosome. The two break at corresponding points, switch fragments, and rejoin. The result is two recombinant chromosomes, and the bacteria can be called a recombinant cell, just like the pencil can also be called a recombinant pencil. The recombinant pieces left outside the chromosome will eventually be degraded or lost in cell division. So the pieces of the pencil that fall into the ground will be lost. The chromosome segment gets into the cell by genetic transfer. Now, what is genetic transfer? Genetic transfer is a mechanism by which DNA is transferred from a donor to a recipient. You transfer DNA from a donor to a recipient. Once donor DNA is inside the recipient, crossing over can occur. The result is a recombinant cell that has a genome different from either the donor or the recipient. In bacterial, genetic transfer can happen in three ways: transformation, transduction, and conjugation. A recombination event must occur. That's why you see I've taken time to explain recombination. Go over again and understand. Once you understand the combination, the other things we're talking about will become easy. Are you following now? A recombination event must occur after transfer in order that the change in the genome be heritable based on the next and pass on to the next generation. Now let's go on to transformation. What is transformation? After death or cell lysis, some bacteria release their DNA into the environment. Other bacteria generally of the same species can come into contact with these fragments, take them up and incorporate them into their DNA by recombination. This method of transfer is the process of transformation. Any DNA that is not integrated into the chromosome will be degraded, will be lost to the environment. The genetically transformed cell is called a recombinant cell because it has a different genetic makeup than the donor and the recipient. All of the descendants of the recombinant cell will be identical to it. In this way, recombination can give rise to genetic diversity in the population. Now, let's talk about the Griffith experiment. Griffith experiment, the transformation process was first demonstrated in 1928 by Frederick Griffith. There's no how we talk about transformation that you will not answer questions about Griffith. So go and put that at the back of your mind. You are going to be asked some questions about Griffith. Griffith experimented on Streptococcus pneumoniae, a bacteria that causes pneumonia in mammals. When he examined colonies of the bacteria on petri dishes, he could tell that there were two different strains. The colonies of one strain appeared smooth. Later analysis revealed that this strain has a polysaccharide capsule and is virulent. That is, it causes pneumonia. The colonies of the other strain appear rough. This strain has no capsule and is avirulent. It does not cause pneumonia. When Griffith injected living encapsulated cells into a mouse, the mouse died of pneumonia, and the colonies of encapsulated cells were isolated from the blood of the mouse. When living unencapsulated cells were injected into a mouse, the mouse remained healthy, and the colonies of non-encapsulated cells were isolated from the blood of the mouse. When living non-encapsulated cells were injected into a mouse, the mouse remained healthy, and the colonies of the non-encapsulated cells were isolated from the blood of the mouse. Griffith then killed the encapsulated cells and injected them into a mouse. The mouse remained healthy, and no colonies were isolated. The encapsulated cell lost the ability to cause the disease. However, a combination of it killed encapsulated cells and living non-encapsulated cells did cause pneumonia, and colonies of living encapsulated cells were isolated from the mouse. How can a combination of these two strains cause pneumonia when either strand alone does not cause the disease? This is because of a combination, and this is transformation. If you guess the process of transformation, you are right. The living non-encapsulated cells came into contact with DNA fragments of the dead encapsulated cells. The genes that code for the capsule, THR capsule, enter some of the living cells, and the crossing over event occurred. The recombinant cell now has the ability to form a capsule and cause pneumonia. All of the recombinant offsprings have the same ability. That is why the mouse developed pneumonia and did what? And died. That is the Griffith experiment, and is used to explain transformation. And transformation is a product of recombination. Even the Griffith experiment is explaining gene transfer and transformation. You are going to be asked to explain the Griffith experiment and how it relates to transformation. Go and note that down. Transduction. Another method of genetic transfer and recombination is transduction. This method involves the transfer of DNA from one bacterium to another with the use of a bacteriophage. A phage is a virus that infects bacteria. The phage T4 and the phage lambda, for example, both infect E. coli. The phage reproductive system is important to understanding transduction. Phages are obligatory intracellular parasites and must invade the whole cell in order to reproduce. In fact, most viruses are parasites. Most viruses are obligatory intracellular parasites. That's why when the virus appears on the surface of a wood or of iron or on the soil, they appear as non-living, they appear as crystals. But when they find a host, suitable host, they take over the system of the host and they start living. So phages are obligatory intracellular parasites and must invade the whole cell in order to reproduce. T4 phage multiplies by the lytic cycle, which kills the host, and lambda multiplies by the lysogenic cycle, which does not cause the death of the whole cell. In lysogeny, the phage DNA remains latent in the host until it breaks out in a lytic cycle. Now, let's talk about the general steps of the lytic cycle. Number 1 is attachment of T4 to receptors on E. coli cell wall. Penetration of the cell wall by take-all, and then this injects DNA into host. Penetration E. coli DNA is hydrolyzed. Phage DNA directs biosynthesis of viral parts using the whole cell's machinery. The phages mature. During the whole cell machinery, the phages mature as the parts are assembled. Lysis of E. coli and release of the new phages then occur. That is the steps involved in lytic cycle. You'll be asked for this step. General steps of the lysogenic cycle. Phage attaches to E. coli and injects DNA into E. coli. Phage circularizes and can enter either the lytic or the lysogenic cycle. The lytic cycle would occur as previously described and lead to the death of the bacterium. In the lysogenic cycle, the circular phage DNA recombines with E. coli DNA, and the phage DNA is now called prophage. E. coli undergoes cell division, copying prophage and passing to daughter. With more divisions, there are more cells with the prophage. The prophage may exit the chromosome and start a lytic cycle at any time. Now that you have seen the phage life cycles, we can discuss transduction in detail. Transduction can be generalized or specialized. What are the steps of general transduction? A phage attaches to cell wall of bacterium and injects DNA. A bacterium chromosome is broken down and biosynthesis of phage DNA and protein occurs. After this occurs, what next? Sometimes bacterial DNA can be packaged into the virus instead of phage DNA. Can you imagine? This phage is defective, can't destroy another whole cell because it does not carry its own genetic material. The cell lysis releases viruses. The phage carrying bacterial DNA infects another cell. Crossing over between donor and recipient, DNA can occur, producing a recombinant cell. Now, in generalized transduction, any bacterial genes can be transferred because the host chromosome is broken down into fragments. Whatever piece of bacterial DNA happens to get packaged within the phage is the genetic material that will be transferred between cells. In specialized transduction, on the other hand, only certain bacterial genes can be transferred. These genes, as you will see, must exist on either side of the prophage. Specialized transduction requires a phage that uses the lysogenic cycle for reproduction. The steps in specialized transduction are as follows. Remember that in lysogenic cycle, phage DNA can exist as a prophage integrated in the bacterial chromosome. Occasionally, when the prophage exits, it can take adjacent bacterial genes with it. The phage DNA directs synthesis of new phages. The phage particles carry phage DNA and bacterial DNA. The cell lyses, releasing the phages. A phage carrying bacterial DNA infects another cell. The joint phage and bacterial DNA circularize. Along with the prophage, bacterial DNA integrates with the recipient chromosome by a crossover event. This forms a recombinant cell. Now we'll move on to conjugation. This is a third mechanism by which genetic transfer takes place. This mechanism requires the presence of a special plasmid called the F plasmid. Therefore, we will briefly review plasmid structure before continuing. What are plasmids? Plasmids are small circular pieces of DNA that are separate and replicate independently from the bacterial chromosome. Plasmids contain only a few genes that are usually not needed for growth and reproduction of the cells. Just for information, this course previously used to be taken as theory. Later, when CBT came, we took it as CBT because about more than 400 students are taking it. Now CBT have said that you can't do 300 and 400 level courses, must be written. Therefore, some of these terminologies in this course, you are going to define them, you're going to explain them. And you explain them in detail, not just explaining them peripherally. So I would suggest that you read more material on this subject so that you can be able to discuss them with more authority. Plasmids contain a few genes that are usually not needed for growth and reproduction of the cell. However, in stressful situations, plasmids can be crucial for survival. The F plasmid, for example, facilitates conjugation. This can give a bacterium new genes that may help it survive in a changing environment. Some plasmids can integrate reversibly into the bacterial chromosome. An integrated plasmid is called an episome. Bacteria that have a F plasmid are referred to as F plus or male. Those that do not have an F plasmid are F minus or female. The F plasmid consists of 25 genes that mostly code for production of sex pillar. A conjugation event occurs when the male cell extends his sex pillar and one attaches to the female. This attached pilus is a temporary cytoplasmic bridge through which a replicating F plasmid is transferred from the male to the female. That is just like a male transferring his sperm to the female, but no, that is not what is happening specifically here. When transfer is complete, the result is 2 male cells. The F plasmid can behave as an episome. When the F plus plasmid is nucleated within the bacterial chromosome, the cell is called an HFR cell, high frequency of recombination cell. The F plasmid always inserts at the same spot for a bacterial species. The HFR cell still behaves as a F plus cell, transferring F genes to a F minus cell, but now it can take some of the bacterial chromosome with it. Replication of the HFR chromosome begins at a fixed point within the F episome, and the chromosome is transferred to the female as it replicates. Movement of the bacteria usually disrupt conjugation before the entire chromosome, including the tail of the F episome, can be transferred. Therefore, the recipient remains F minus because the F plasmid is not entirely transferred. A crossover event can occur between chromosomes, genes on the HFR fragment, and the F minus DNA. Pieces of DNA not recombined will be degraded or lost in cell division. Now, the recombinant genome can be passed on to future generation. That is conjugation. I want you to take note of all these special terms and explanations and mechanism of action. Now we'll move on to talk about plasmids as a standalone quickly. Plasmids, as we've said, are genetic elements that can also provide a mechanism for genetic change. Plasmids are small, circular pieces of DNA that exist and replicate separately from the bacterial chromosome. You have already seen the importance of the F plasmid for conjugation, but other plasmids of equal importance can also be found in bacteria. One such plasmid is the R plasmid. R plasmid, also called resistance plasmid. They carry genes that confer resistance to certain antibiotics. A R plasmid usually has 2 types of genes: R determinant, resistant genes that code for enzymes that inactivate certain drugs. Then the RTF, resistance transfer factor, genes for plasmid replication and conjugation. Without resistant genes for a particular antibiotic, a bacterium is sensitive to that antibiotic and probably destroyed by it. But the presence of resistant genes, on the other hand, allows for their transcription and translation into enzymes that make the drug inactive. Resistance is a serious problem. The widespread use of antibiotics in medicine and agriculture has led to an increasing number of resistant strain pathogens. These bacteria survive in the presence of the antibiotic and pass the resistance genes on to future generations. R plasmids can also be transferred by conjugation from one bacterial cell to another, further increasing numbers in the resistant population. So we're talking about plasmids, that they are small circular pieces of DNA that exist and replicate separately from the bacterial chromosome. And we said that there are, apart from the F plasmid, which is used for conjugation, there are also R plasmids. R plasmids carry genes that confer resistance to certain antibiotics. it makes certain antibiotics to be resistant. And there are two types: the terminal resistant genes and the resistant transfer factor. And then without resistant genes for a particular antibiotic, a bacterium is sensitive to that antibiotic and probably destroyed by that antibiotic. So because of resistant plasmids is one of the factors why we have the resistant plasmid is a sort of gene modification that confers antibiotic resistance on an antibiotic, on a compound, on a drug, and on a bacterium, and then it inactivates the drug. So there is a bacterial infection, you use an antibiotic, the bacterial infection still remains, the antibiotic, and there's resistance. It can be due to this R plasmid, but not only R plasmids. And because of these, new antibiotics are to be developed. We have the first generation of antibiotics. Later, resistance came. Resistance can be due to so many factors. One of them, which is key to us here, is the resistance plasmids. Antibiotic resistance can also be caused by drug abuse. It can be caused by misuse of drug. It can be caused by the target site of the drug not available. What the drug is targeting not being available. Like, let's assume a drug is targeting the cell wall of the bacterium, and a bacterium that has no cell wall, like mycoplasma, is the cause of the infection. That antibiotic or drug that is targeting the cell wall is not going to work. It's not going to function. So today we have up to 6 generations of antibiotics, and I think more antibiotics are being developed. Transposons. What are transposons? Transposons are transposable genetic elements, and they are pieces of DNA that can move from one location on the chromosome to another location, from plasmid to chromosome, or from chromosome to plasmid, or from one plasmid to another plasmid. The simplest transposon is an insertion sequence. And the second insertion sequence contains only one gene that codes for transposase, the enzyme that catalyzes transposition. The transposase gene is flanked by 2 DNA sequences called inverted repeats, because that 2 regions are upside down and backward to each other. Transposase binds to these regions and cause DNA to remove the gene. The transposon can enter a number of locations. When it invades a gene, it usually inactivates the gene by interrupting the coding sequence and the protein that the gene codes for. I'm happy that you have looked at sequences. It's like you have a sequence of 1 to 10 or 1 to 100, and then transposase transpose itself between 10 and 20. After 10, are you going to jump to 20? No. These transposase that have come, the transposon that have entered a number of locations, is going to prevent your easy reading of the sequence, and that happens also to nucleotide sequences. So transposase binds to these regions and cuts DNA to remove the gene. The transposase gene is flanked by 2 DNA sequences called inverted repeats, because that 2 regions are upside down and backward to each other. An insertion sequence contains only one gene that codes for transposase, the enzyme that catalyzes transposition. The transposon can enter a number of locations. When it invades a gene, it usually inactivates the gene by interrupting the coding sequence and the protein that the gene codes for. So the moment a gene is 1 to 10 and then something is between 5 and 6. 1, 2, 3, 4, 5. It is not going to be 6, it's going to be 1, 2, or 5. A, B, C, D, E, F, G, H. A, B, C, D, A, B, C, D, E, G, F, G, H. M, N, O, P, Q. Then are you going to just continue seeing 6 to 10? Normally 1 to 10 is a series. 1, 2, 5, 7, 8, 9, 10. But now 1, 2, 3, 4, 5. A, B, C, D. B, C, D, B, C, D, E, F, G, H. And now say 5, 6, 7, 8, 9, 10. It's not going to flow. It's not going to give us the value that it means. Because that arrangement 1, 2, 3, 4, 5, 7, 8, 9, 10 has meaning. The codon, three base pairs forming a codon, they mean something. So anything that comes in between the reading frame is going to change the whole material. So when it invades a gene, it usually inactivates the gene by interrupting the coding sequence and the protein that the gene codes for. Luckily, transposition occurs rarely and is comparable to spontaneous mutation rates in bacteria. This is even good. We are going to also talk about mutation very soon. So transposition does not usually occur, just like mutation does not usually occur in nature, but it occurs one mutation in a million. One mutation in a million cells occur. Mutation does not occur every time. So complex transposons consist of one or more genes between two insertion sequences. The gene coding for antibiotic resistance, for example, is carried along with the transposon as it inserts elsewhere. It could insert in a plasmid and be passed on to other bacteria by conjugation. This is where we are going to draw the line on the subject of methods in microbial genetics. Like I said when I started, these methods cannot be ended. I cannot exhaust the methods. So you have to take your time, look into the material. I've made explanation, check materials. I'm going to share some diagrams with you. Look at those diagrams, understand them, look for other diagrams for yourself, get other materials on your own, and go in depth into this subject, because no singular individual can be able to complete and know all of the methods in microbial genetics. So I'm going to talk to you some other time where we'll look about replication and also talk about mutation. And for now, see you later. Bye-bye.